Meet Lucy, a 3.2 million-year-old relative of ours who lived in what is now Ethiopia. In 2022, scientists announced that they'd successfully extracted DNA from her bones. This isn't the first time that ancient DNA has made headlines. But when it comes to this field of science, there are some important details you might not know. Like, how long can DNA survive in the first place? To find out, let's start with a little science. DNA is a molecule that contains our genetic information. It's like an instruction manual for building and maintaining a human body. And just like any instruction manual, it needs to be kept in good condition if you want to follow it. Unfortunately, DNA isn't built to last. Most of the time, when we think of DNA, we're imagining the double helix structure, a ladder twisted into a spiral shape. The rungs of this ladder are made up of four chemical bases: adenine, guanine, cytosine, and thymine, or G, C, and T for short. These bases pair up with each other in a very particular way. Adenine always pairs with thymine, and guanine always pairs with cytosine. The order of these bases along the ladder holds our genetic information. So when cells need to use the instructions in our DNA, they read the sequence of bases and translate that into a set of instructions for making proteins, the workhorses of our cells. But DNA isn't always perfectly paired. Sometimes one of the bases gets substituted for another. For example, imagine a sequence of a CGT. If cytosine gets substituted, it could become a CGU instead. That might seem like a small change, but it could have serious implications. After all, that one base determines which protein the cell makes next, which could be something as important as an enzyme that helps break down toxins in the body. Substitutions like this can happen on their own due to mistakes during replication, but they can also be caused by things like UV radiation or chemicals. And over time, these substitutions accumulate until the DNA molecule breaks apart completely. So how long does that take? It depends on a few factors like temperature and humidity. Studies have found that under really hot and dry conditions, DNA can last for hundreds of thousands of years. On the other hand, under wet and humid conditions, it tends to break down much faster. On average, scientists estimate that DNA can survive somewhere between 1 and 500,000 years. But most of the time, it doesn't make it that far back. The vast majority of ancient DNA found at archaeological sites is less than 100 years old. And while our DNA has a limited shelf life, it's still pretty tough. In fact, it's surprisingly resilient to some types of damage. When exposed to UV radiation, for instance, the bonds between bases on either side of the molecule get damaged at about the same rate, meaning that even if one strand breaks, the other strand can often repair it. But there's another kind of damage that can be caused by the same source, UV radiation, that doesn't play by those rules. You see, if a molecule of water on the surface of a cell gets hit by UV radiation, it can split apart into two parts, a hydroxyl radical and a proton. Hydroxyl radicals are highly reactive, so they'll quickly try to stabilize themselves by snatching electrons away from nearby molecules. And guess what happens to be a great source of unprotected electrons? Unpaired bases. So when this happens, a single UV photon can cause multiple substitutions in a row, and those substitutions tend to destroy whatever gene they occur in. Luckily, cells have a mechanism for dealing with this type of damage, but it takes time to kick in. And if UV exposure is heavy and prolonged, like during an intense volcanic eruption, for instance, cells may not be able to keep up. So genes can get destroyed before they have a chance to be repaired. Luckily for us, this sort of damage is rare in the fossil record. Most fossils form under conditions that prevent these kinds of molecular shenanigans, which is why DNA can survive in them for so long. But it's a different story for organisms that live most of their lives in the water. Marine organisms are constantly bombarded by UV radiation because water doesn't block it very well. Plus, it's hard for marine organisms to keep their DNA protected. They can't slather themselves in sunscreen, So many of them have evolved to repair damaged DNA more efficiently. But even that's not enough to save their DNA from breaking down completely. As a result, almost no ancient DNA has been found in marine organisms older than 100,000 years. Speaking of marine organisms, let's return to Lucy. Lucy was an early relative of modern humans who lived in what is now Ethiopia. Because she was on land rather than in the sea, her DNA had a much better shot at surviving. But it still took a combination of extremely dry and cold conditions to preserve her DNA for 3.2 million years. Conditions like the ones found in the mountains of Ethiopia. Thanks to these conditions, Lucy's bones were able to protect her DNA for much longer than it would have survived in most other environments. Even so, the extraction process was fraught with difficulties. Her fossilized bones had to be carefully cleaned and crushed into a fine powder. Then the researchers used a combination of physical and chemical methods to isolate the tiny amount of DNA present in each sample. In the end, they managed to extract about 111 million base pairs. And while that sounds like a lot, it's actually only about 0.1% of the total genetic material present in a single cell. So even though Lucy's DNA had survived for millions of years, the amount of it present in her bones was extremely degraded and incomplete. What's more, Lucy's DNA wasn't exactly in great shape. Over the course of millions of years, bacteria had colonized her bones and filled them with their own DNA. Plant matter and soil had worked its way into her skeleton, adding even more contaminants. And that is not even mentioning the DNA from the scientists themselves that inevitably ended up in the samples. All of this contamination made it impossible for the team to figure out where the bacterial, plant, and human DNA began and ended. So they had to develop new computer algorithms to sift through the mess. And after all that, the team still couldn't reconstruct the entire genome. The best they could do was identify about 1 point per million of the original to point per million base pairs that make up Lucy's genome. So while scientists have managed to find some pieces of Lucy's genome, they've only been able to piece together a fraction of her genetic code. In fact, reconstructing even a partial genome from such degraded material is considered to be pretty ambitious. Scientists have typically focused on finding much larger pieces of DNA. For instance, in 2003, a group of researchers announced that they managed to find 100,000 base pairs of DNA in a fossil of a Neanderthal relative named Oase1 that was about 40,000 years old. Similarly, in 2019, another team announced that they managed to recover 2.6 million base pairs of DNA from a Neanderthal bone that was only 39,000 years old. Both of these studies were able to analyze much longer sequences of DNA than the Lucy study because the DNA hadn't had as much time to degrade yet. So given the difficulties of studying degraded DNA like Lucy's, you might be wondering, can we ever reconstruct a full dinosaur genome? Short answer is probably not. Longer answer is probably not without some major scientific breakthroughs. The oldest DNA that scientists have ever recovered is about 1.2 million years old. But since dinosaurs went extinct about 66 million years ago, their DNA has had a lot more time to degrade. Plus, they would have been mostly aquatic animals, so their DNA would have broken down even faster. Even if we did manage to find a few pieces of dino DNA, we have to contend with another problem. The DNA of modern organisms, like us and dinosaurs, probably diverged between 300 and 600 million years ago. So even if we had a complete dinosaur genome, we'd still need to compare it to the genomes of several other extinct species to fill in the gaps and figure out which genes were unique to dinosaurs. Without that information, we wouldn't really learn anything new. Of course, scientists never stop pushing the boundaries of what's possible, so maybe we'll figure out a way to study ancient DNA someday in the future. But for now, it seems like dinosaurs will remain a little bit more mysterious. Thanks for watching this episode of SCI Show, and thanks especially to our pandemonium of padrons. Our podrons are the reason we can make videos like this. So if you'd like to help us make more amazing science videos, you can go to our support page to learn more.